Okay, so welcome back to this next video in which we are discussing the life cycle of HIV. Okay, so we've discussed that HIV has a viral envelope, which is a stolen piece of phospholipid bilayer that the HIV virion particle took from the cell that it budded off, basically. Excuse me. And then underneath that viral envelope, what you have is a spherical surface of P17 matrix. Okay, so this is a spherical protein surface that is made out of loads and loads of P17 proteins. Within that matrix, what you then have is this capsid, which is in a cone shape and is constructed out of P24 proteins, and within the capsid what you have is the genetic information, which is in the form of single-stranded positive sense RNA. Okay, basically what this means is that these pieces of RNA could actually be read by ribosomes and used to make proteins directly. Okay, now that's not actually going to happen in this case. What's going to happen for human immunodeficiency virus is that the DNA is first going to be the RNA is first going to be turned back into DNA, and then the DNA is going to be implanted into the human genome. Okay, but in principle it could be translated directly. Okay, so I hope you appreciate that if you've got this positive sense piece of RNA, you could make not only the negative sense piece of RNA, but you could also make all of the DNA back again. You could first synthesize the piece of DNA that's complementary to this, the coding strand, and from the coding strand you could certainly synthesize the non-coding strand. Okay, so this contains all of the genetic information, basically. It's just a different way of storing it, basically. Now, also in the capsid, you have 3 really important enzymes that the virus brings with it because the host cell just does not have them, basically. Okay, so where should I draw these? So I'll put 1 in here, 1 here, and 1 here. So these are 3 really important enzymes. So I'll give them all a separate little colour. So I'll colour in this first one in orange. Now this one that I've just coloured in orange, this can represent the reverse transcriptase enzyme. Okay, and this is the enzyme that's going to take the piece of positive sense RNA and convert it back into a piece of DNA, basically. Reverse transcriptase. Okay, so reverse transcriptase is there in orange, and normal human cells do not have reverse transcriptase. We do not need to be able to turn mRNA back into DNA, basically. Okay, so the next one along, the one in the middle, can represent integrase. Now this is the enzyme that will implant the DNA, once we have produced it, back into the human genome, basically, or rather into the human genome, because it hasn't been in there before. Okay, so it's going to implant the viral DNA that we're going to synthesize into the human genome. Okay, and then finally, what colour should I colour this final one in? Yellow, maybe. Okay, finally, this is the protease. Okay, and this is essential for altering the proteins that the viral DNA is going to produce, basically, because what happens in the viral production of proteins is that you produce polyproteins, and this concept is that basically you synthesize loads of different proteins all as one great big polyprotein. Okay, so you make a massive great polypeptide that actually has loads of different proteins that you want to be separate within it, and then you'll cut it up into smaller sections, and all of those different sections will become their own proteins. So the concept of a polyprotein is that you start off with a massive great polypeptide, like so. Okay, so this is a polyprotein, and you're then going to cut it up into different bits, and then each of these bits is then going to become its own protein, basically. Okay, so you need some enzyme that can cut the polyprotein into these different bits, and this is the HIV protease enzyme, and that's in yellow, and the virus again brings that with it. Right, okay, so now we've discussed the structure of HIV. What I now want to discuss is the genome of HIV in a little bit more detail. So I want to discuss what genes does HIV actually have. Now, this is where another little bit of complicated genetics comes up, and this really is the most conceptually difficult bit of this entire video, basically, because the HIV genes, the different genes, are not just one after the other. So if I take this piece of mRNA here, okay, so let's say this is our single-stranded positive sense piece of mRNA, RNA from the HIV virion. And let's say this is the 5' end, and this is the 3' end. Now, what you might like to think of is that you have one gene here, let's say, and then a little bit downstream of that you might have another gene here, and then maybe a little bit downstream of that you might have another gene here. What you would not like to think about is that maybe the genes could be overlapping. Maybe you could have a gene that was here. That would seem a little bit odd, but basically this is the way the HIV genome actually works. It has genes which overlap with other genes. Now you might ask, how does this work? You might think, well, okay, obviously these two proteins that we're going to get here, they have this shared portion, okay? So they'll have an identical portion of amino acids, but it's not like that. These two proteins will not have a shared portion of amino acids, and the reason is that they will be read in different frames, basically. Okay, so to try and explain this, let me go back to my central dogma of biology, and we're going to need to look at the process of translation in a bit more detail. So we've already sort of looked at the core idea, which is that basically triplets of organic bases will code for a single amino acid, and that's the crux to all of this. Okay, so if we take our piece of mRNA here, let's draw this picture a little bit bigger now. So here's our 5', here's our 3' end. Then basically the RNA is just a sequence of organic bases. So it's just organic base after 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 organic base. Now, when the ribosome reads the RNA, how does it decide how does it decide which reading frame to go in? Okay, and let me explain this, what I mean here. So basically we've discussed that there is the triplet code, okay? So basically it's not just the case that one organic base on the RNA codes for one amino acid. Instead, 3 of them together will code for one amino acid. So what we could do is, if we wanted to read this piece of mRNA, we could start here. We could look at this codon here. Okay, so the name for a triplet of organic bases like so is called a codon. Okay, because it will code for a single organic base. Okay, you will look at this combination of 3 organic bases, and that will code for a single amino acid. Okay, so there's some amino acid basically. Okay, so I'll just put that there. There's a specific amino acid for that codon. Then you will go to the next codon along, the next 3 here, like so. Okay, and you will look at what amino acid that codon codes for, and you will join this amino acid to the next one along. Okay, and then you'll go on further. So here's another codon here. You will look at which amino acid this codes for, you'll put that in as your 3rd one, and then you'll go on like so. Okay, codon after codon after codon, and you'll put amino acid after amino acid after amino acid. But think about this. What would happen if I now shifted all of the codons along by just one organic base? So let me explain what I mean by this. Let me draw this picture again. I think I'll draw it again rather than trying to superimpose what I mean on this. Okay, so I need the same organic bases again. So we've got 2 here and then 12 following it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so initially what we did is we started with this codon here. Okay, that's that codon here. But what if instead of starting with those 3, we'd instead started, and let me draw this carefully, we want this one here. and this is going to become a mess, but never mind. Okay, so what if instead we'd started with this codon here? So I've removed that one amino acid there, but now I've got not amino acids, organic bases. I've removed that organic base there, but now I've got these 3 organic bases here as this codon, and then I'll continue on. So I'll get this codon coming after it, like so. Okay, and this codon coming after it here. Okay, this is the concept of a different reading frame. Okay, so basically I am now in a different reading frame. The codons that I get here will be completely different to the codons that I get here, and the amino acids that you'll be putting in are completely different. So basically, by shifting along by one organic base, I have completely changed my reading frame. So that's the concept of a reading frame. It means how are you choosing the 3 organic bases that you want to use as a codon. I don't really know how to define it. It is this. This is what it is. It basically means that how are you clustering the 3 organic bases together to make a codon. Here we picked this 3rd organic base here, 4th and 5th, to make a codon. Here we've picked the 4th, the 5th and the 6th to make a codon. It means how are you clustering them together to make codons. And as soon as you've selected the first one, then all of the reading for all of the other codons is then set, because you just go on to the next codon and then the next codon. So there are in total 3 different reading frames that you could use. You could use this one that I've drawn out here, this is number 1, then you could use number 2 here, but there's also a final one, which is where I've shifted 2 organic bases down. Okay, so this one would be the first organic base of my first codon, and then it would take the 2 from here. So let's just draw that out as well. Okay, so basically for any piece of RNA, there are 3 plausible reading frames, basically, that the ribosome could use. 1, 2, 3, how many did we say we have? 14 in total. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, so this time what we'll do is we'll skip down 2, so we'll skip the first 4, and then we'll use these 3 here as our first codon, and then you'll continue on to your next codon, and then your next codon still. Okay, so that again is another reading frame, and this will result in completely different amino acids again. So it will result in a completely different protein if you read in that reading frame. So the same piece of RNA could actually be read in totally different ways and could produce a totally different sequence of amino acids, basically. And this is how this overlapping is going to work. So basically, we will be reading the RNA in different reading frames, and that's how we'll get totally different sequences of amino acids in our proteins, even though we're using the same combination of amino acids. So basically, when I have these 2 overlapping here, I am not going to be reading this overlapping portion in the same reading frame for, let's say, gene 1, which is this one here, and gene 2. So we'll be reading this overlapping portion of RNA here in different reading frames, so we'll be getting totally different sequences of amino acids put in. Okay, so now with this understanding, we are in a position to look at the actual genes in the HIV genome. Okay, so some of these we will actually see the function of, the main ones, but I'm going to give you all of them. Some of them we're not going to touch on because they're a little bit more trivial, basically. We'll look at the main ones. So, basically, the HIV genome starts with what is known as the LTR, okay, and this stands for long terminal repeats. Okay, so this stands for long terminal repeats, and let's say this is the 5' end, and this long terminal repeats here will actually contain the promoter region which controls the transcription of the entire HIV genome. Okay, so this of course will be once we've got the DNA rather than the RNA, but at the moment we're looking at the RNA at the moment. Okay, then downstream of the long terminal repeats, what you have is a special gene known as GAG, And this one we will most definitely see the function of this. Okay, then after GAG, what you have is another important gene, and there's an overlap here. Okay, so the two are overlapping. Okay, and this means that we will be reading this overlapping portion of organic bases in different reading frames, basically. Okay, and this is the POL gene. Okay, so there's GAG and POL. After that, we have another gene, and again it overlaps with POL, so again it will be in a different reading frame, and this is VIF. Again, we'll see the function of VIF later. Okay, another gene that again overlaps with VIF. Okay, and I'm worried now that we're going to run out of space because I've been drawing these so large. And the next one is called VPR. Okay, then a little bit more downstream, and I'm going to try and draw things a little bit smaller now so that we can actually fit things in. Okay, the next little gene we have is known as VPI. Okay, and we also have a little gene in here. Okay, like so, which is called TAT. And overlapping with TAT, we also have a little gene called REV. Now, you have two of each of these genes, so I won't label them up yet, because you're going to have another one further down. Okay, then here, another really important gene, ENV. This is going to code for the viral envelope proteins, and I've just realized that we need to go back to our structure of HIV because I have missed off the viral envelope proteins. Okay, we'll come back to that in a moment. Then you have another little gene here called NEF, and now I'll put in the other TAT and REV genes, so you also have another TAT gene underneath here, and then another little REF gene underneath here. Okay, so these are REV genes here, this one, and these are TAT genes here, 1, 2. Okay, and we're not going to touch on what these actually do. Okay, and then finally you have another long terminal repeat region right at the end, and it overlaps with the NEF gene. And then we'll have the 3' end over here. Okay, so that's the structure of the genome of HIV. Now we'll look at some of these genes as we go through, specifically GAG, POL, ENV, and VIF. We will look and see what those do. So, if you want to forget about these other ones, you can do. GAG, POL, ENV, and VIF are the only ones we're actually going to see what they do. Right, so before we go on, I just want to go back to my structure of HIV and add in something very important that I have forgotten, okay, and that's the proteins that are within the viral envelope membrane. Okay, so you have two important proteins that are basically complexed together. Okay, so this first one that I'm drawing is GP41, and this stands for glycoprotein 41. So the GP stands for glycoprotein. Okay, so glycoprotein 41, and then bound over the top of glycoprotein 41, which I'll draw like this, you then have another one, and this is called glycoprotein 120, GP120. Okay, and the viral envelope will be covered in these complexes. So here's another one, here's another GP41 with GP120 bound to it. I'll put another one over here. So you'll have absolutely hundreds of them, well, more than hundreds. Okay, you'll have a huge number of these all over the viral envelope. Okay, and these are going to be extremely important for the infection process of the HIV virion particles into the cell. Okay, right. So, what we're now going to do before we finally go on to the actual infective life cycle, i.e. how it infects cells, is we're going to talk about some of the divisions of HIV-1. Okay, so we've discussed that there are these two major divisions of HIV. So firstly, I should just say that HIV is one species of virus, okay, but in there are absolutely loads and loads of genetically distinct strains, okay, and they can be broadly divided into these two categories of HIV-1, which is the more common, and then HIV-2. Okay, we are completely focusing on HIV-1 in this video. Okay, now HIV-1 strains of HIV-1 can also be further divided into 4 separate families, basically, or 4 separate groups. So there is the M group, okay, which stands for the major group. Okay, there is then the N group, which stands for neither group. Okay, but this is actually quite a common, well, it's a more common group than the other two we're about to see, neither group. Okay, and I wish I'd spaced it out a little bit more now. Okay, so this next group is called the O group, which stands for outlier group. Okay, so really it was the major group, the outlier group, and then the group that was in neither. But then in 2009 they created a final group, well, they found another one, and this is the P group. Okay, right, so this is only since 2009 that they've had this P group. Right, so by far the most important of these 3 groups is the major group. So we're going to ignore the other 3 groups and just talk about the major group. So within the major group there are 9 different genetically distinct strains of HIV, and these are often called the clades. Okay, so you can split M into the different clades, and these are all genetically distinct. So there is the A clade, the B clade, the C clade, and then it goes on all the way down to K.